Hello and welcome to Alexander Disease Research Update, episode number 26, recorded on March 25th, 2025. I'm Al B. Messing from the Weissman Center at the University of Wisconsin, and with me today are Tracy Hagemann, also from Wisconsin, and Rob Berman from the University of California, Davis. Tracy's been on before, but Rob, since this is your first time on the podcast, and I know you'll be on again, can you say a bit about your background and why Tracy and I recruited you to work with us on Alexander disease?
Okay. Well, I'm a behavioral neuroscientist at the University of California, Davis. I use animal models to study neurological diseases. I've been working in the field for now over 40 years. And I'm interested in neurodegenerative and neurodevelopmental disorders. I was directing a research laboratory that has the technology to study complex behaviors in rats. And Albie and I and Tracy got together and decided that it would be a good thing to use the facilities that I have at UC Davis to try to characterize their new rat model of Alexander disease. So basically my interests and their interests overlapped, and so we began this study. Now it's been, 10 years?
Close to it.
It's taken a while, yes. That's one of the key messages for today. These things take time. Well, thank you both for being here. Before we get started, please send feedback and questions to my email, which is amessing@wisc.edu. So today we're going to talk about a paper of ours that has just come out entitled GFAP Mutation and Astrocyte Dysfunction Lead to a Neurodegenerative Profile with Impaired Synaptic Plasticity and Cognitive Deficits in a Rat Model of Alexander Disease, which is now in press in the journal eNeuro. Since I'm one of the co-authors on this paper, I figure I could start off by briefly stating what I viewed as the goal of this study, which is to switch from what's been our longtime focus on astrocytes to the other cells of the central nervous system. Of course, GFAP is the causative gene in which variants cause Alexander disease. GFAP is almost exclusively expressed in astrocytes, but we know that these changes in astrocytes impact other cells. First of all, oligodendrocytes. Of course, it's a leukodystrophy. We've talked before about microglia, but the fourth cell type, major cell type in the central nervous system, are the neurons. So in this paper, we're switching our focus from astrocytes to neurons and trying to examine how the GFAP mutations affect neuron gene expression and the connections between neurons, their synapses. So the first part involves looking at changes in gene expression, and in the second part, we look more specifically at the effects on neurons, both in terms of neuronal number and secondly in terms of neuronal function, especially as it relates to behavior and specifically the cognitive behavior. So why don't we get started with you, Tracy? The title of this paper includes the phrase neurodegenerative profile. What exactly do we mean by that?
Well, I think if you look at gene expression patterns in other diseases that people typically think of as being chronic diseases of aging, like Alzheimer's and Parkinson's, there's a profile which includes neuroinflammation, so increased expression of genes related to an immune or stress response, and then a decrease in expression of genes related to synapses and synaptic function, and also metabolism and mitochondria. So the powerhouse of the cell seems to have some dysfunction in some of these diseases.
So how did you assess potential changes in gene expression?
Well, we used what many people think of as an unbiased approach to look at gene expression from total RNA, which is the mediator between DNA, so your genetic makeup, and the synthesis of proteins, to look at the expression of all genes in a particular region. We chose hippocampus, which is a region of the brain that controls memory and learning. And the reason we did that is because we see a lot of GFAP accumulation in that region, particularly in our rodent models. There's also pathology in the hippocampus in the human disease. So we focused on hippocampus, and we extracted RNA, and then we looked at total gene expression. And so the first part of the paper describes what we found, and that was, the title implies, was a neurodegenerative profile. But we also followed that up to look at protein, because just because transcripts are up or RNA is increased doesn't necessarily mean that protein is. So we looked at things like cytokines and chemokines, which would be part of the inflammatory response. We quantified those, and sure enough, we saw several of them were elevated at the protein level in the model, in hippocampus. And we also looked at synaptic proteins, and we saw that those were downregulated along with proteins that are encoded for in mitochondria. So all of those things together correlated with each other.
We should probably clarify that the type of analysis that was done here did not look selectively at individual cell types, rather it was all of them together.
Right. So it would be very interesting to come back and do a similar experiment where we looked at individual cells and the expression profile. So you could separate out things like astrocytes, neurons, and other inflammatory cells like microglia.
So you also compared the gene expression profiles at 2 ages, 3 weeks and 8 weeks. Why did you pick those?
Well, 3 weeks, the animals are quite normal. It's right after they're weaned from their mother, and it's very difficult to distinguish an animal with the Alexander disease mutation from one of its wild-type littermates. But yet they still have a lot of pathology. If you actually look at the central nervous system, they have a lot of GFAP accumulation at that age, and yet they're not showing symptoms. So we thought that was an interesting age to look at because it's more or less pre-symptomatic, and they're not showing behavioral deficits that we've been able to detect. And then at 8 weeks of age, that's probably the most severe time point for the disease in the rodent, in this particular rodent model. And we lose some of the animals. We don't know why they die, but about 15% of the animals die at this period, and they're very frail and gaunt, and they're not doing very well at that point.
So what were the major findings?
Well, at an early age, we see the initiation of an inflammatory response in the central nervous system. And in my mind, this is intrinsic to the central nervous system, so it doesn't necessarily have to do with an infection or anything like that. It's the brain itself mounting a neuroinflammatory response. And we see a progression of that at 8 weeks of age. So many of the same genes that are elevated and highly elevated at 8 weeks are already elevated at 3 weeks, but just not to the same degree. But if you look at the other changes that we see, such as the loss of or the decrease in expression of genes related to synapses or metabolism, that's a little different. You see a little bit of a change at 3 weeks, but many more genes change at 8 weeks, suggesting, to my mind, that the inflammatory response precedes what's happening at the level of the synapse and the metabolic support. It's also interesting, though, that one of the things that we didn't highlight, at least not in the abstract of the paper, was that at 3 weeks of age, we also see changes in expression of genes related to lipid or cholesterol metabolism, which in my mind is interesting because that could contribute to deficits in oligodendrocytes. With the hippocampus, it's not got a lot of white matter in it or myelinated tracts. It has some, but it's not considered a white matter structure. So it just raises the question in my mind of what if we did look at a structure that had more white matter in it, and how relevant would those changes in lipid metabolism be elsewhere in the brain?
So generally speaking, inflammatory markers are going up, synaptic markers are going down,
right,
and genes related to lipid metabolism are going down.
Right, and mitochondrial genes are also going down. Genes that are expressed by nuclear DNA and the mitochondrial genome itself. So at 8 weeks of age, we see a marked decrease in genes expressed by the mitochondria itself.
Yeah, mitochondria are really interesting, and there's a long history of studies of intermediate filaments and interactions with mitochondria. There are some patients with fundamental mitochondrial diseases that have been misdiagnosed with Alexander disease in the past, and so going forward, of course, we'd really like to learn more about how mitochondrial functions impacted in Alexander disease. So you also went on to do counts of neurons. And actually, this is a topic that is a crossover between Tracy's domain and Rob's domain. But I'll let Tracy have a go at it first. How did you do the neuron counts, and what did you find?
The neuron counts were fairly simple approach. We just looked at the nucleus of it with a general marker for neurons, and we counted those through a particular stretch of the hippocampus, and we looked at different regions of the hippocampus trying to narrow down where there may have been deficits. So we looked at three main regions. They're referred to as CA1, CA3, and the dentate gyrus. And we looked at those regions at both 3 weeks and at 8 weeks. We saw no differences at 3 weeks. But at 8 weeks of age, we did see a decrease in neurons, but only in the dentate gyrus. And whether that is reflective of some of the changes that we saw with neuronal genes or synaptic genes, it's hard to say. But one of the findings that we've had in both our rat model and in the mouse models is that we see deficits in what people refer to as adult neurogenesis. And the hippocampus is one of the few places in the brain that you see the production of new neurons. And that specifically happens in the dentate gyrus. And we've previously shown in our mouse models that there is no adult neurogenesis in that region. And the same is true in our rat models. And at this time point, at 8 weeks of age, you see no markers of new neurons. That's not true at 3 weeks. And so maybe that is what's contributing to the changes in the number of neurons, especially at 8 weeks in this region.
Of course, there's still a lot of debate about what this whole phenomenon of adult neurogenesis means.
Right.
It's a very clear finding in the rodent models that it's really impacted. Whether that's really occurring in human Alexander disease, we don't yet know, and what its significance would be, we don't really know yet.
And Rob can probably say more about the, adult neurogenesis in rodents has been correlated with spatial learning. And so I think that was part of our choices with some of the behavior tests that we did as well. But I'll let Rob talk about that because it's certainly not my area.
So let's turn to Rob. So first of all, Rob, before we even get to behavioral studies, we explored some aspects of neural physiology, particularly a phenomenon known as LTP, or long-term potentiation. Can you explain what that is and what we found?
Right. Well, it is a way to study what is thought to be a synaptic mechanism underlying memory at a cellular level. So most of the studies that are done are carried out in the hippocampus, and as Tracy already mentioned, that's one of the central brain regions important for memory storage and for learning in general. So let me start out by saying that there's a lot of evidence now that when you learn something, connections between neurons are strengthened, or we could say they're potentiated. And so what we think is happening during memory is that networks of neurons are forming interrelated connections, and that represents a memory trace. So we can study this artificially, really, in brain slices. So we took slices of brain from the Alexander disease rat model, and we were able to artificially activate connections within the hippocampus and study the strength of connections between neurons. And what we find under normal conditions is that when you stimulate or activate these connections and do it repetitively, you get a much larger response. So it's called potentiation of the synapse, or long-term, because it lasts a long time. What we found in the hippocampus is that the strengthening of the synapse when you activate these connections is weaker than it is in normal animals, and that suggests an underlying impairment in the basic cellular mechanisms that are necessary for memory formation. So again, it's a way to get at actual cellular and synaptic mechanisms of memory that we think are related to some of the behavioral or cognitive deficits we see in the rat model of Alexander disease.
All right, so let's turn to behavior. we focused on cognitive behavior. There'll be a follow-up paper where we focus more on motor functions, but now we're talking about cognitive behavior, and a definition I pulled from Webster's dictionary is this is conscious intellectual activity relating to thinking, learning, and remembering. And of course, rodents clearly learn and they remember things. What it means for a rat to think, I really don't know, but still, we wanted to look at cognitive functioning in these animals because cognitive deficits are clearly a major aspect of the problems in human Alexander disease. So the first test we did was novel object recognition. Rob, how is that test done and what were the results?
Novel object recognition is a visual object recognition memory test. And let me give you an example that you might be able to relate to. If you are in a room you're very familiar with and you know where all of the objects are, the lamps, the table, the flowers, and you're in that room for an hour or two, you leave and you come back and there's a new object there, a brand new vase, for example, you're likely to see that and maybe go over and explore it. And you can do that because you have a memory of the objects that were in the room that are no longer novel, but the new object is something that attracts your attention, and because you don't remember it there before, you go over and you look at it. So it's really a test to see whether or not you're able to recognize that something has changed in your visual world. Okay, so we test that in rats in a box where we put the rats in with two identical objects, and they can explore it for, in our case, for an hour.
What would be an example of?
We have a cone-shaped object that looks like the kinds of cones they put on the street when they're doing construction. They're small. Animals are put in for 30 minutes or an hour, and they explore, but they eventually stop exploring because there's nothing new to see. We take the animal out of the box, we replace one of the objects with a new object. In this case, it was a glass bell jar. Totally different in appearance, but the same relative size. Then we put the rat back in the box and we ask, do you notice that there's a new object in the box? And the way animals have to process that is they have to know one of the objects is one I'm very familiar with. There's no reason to go look and spend much time looking at that object. The new object is pretty interesting. They explore it. So we can look and see that there's an increase in the exploration of this novel object. Okay, so it's a test of visual object recognition. It requires activation of visual areas of the brain, but also an area called perirhinal cortex, which interacts with the hippocampus, and Tracy's already indicated how important the hippocampus is for memory. So our Alexander disease rat model shows poor visual object memory, okay, suggesting that that part of the memory system is not functioning at normal levels.
Okay, so the next test we did was the Barnes maze. What is the Barnes maze?
Well, the Barnes maze is a spatial memory test, and I'll explain what I mean by that. It was actually a test developed by Carol Barnes at the University of Arizona to study the role of the hippocampus in age-related memory problems, and it's become one of the most widely used tests of spatial memory. And basically what we do is we put rats on a platform that has 18 holes. One of the holes is an escape hole. So the animal can be put on the platform, needs to locate and escape through the hole. Okay, they're just escaping a very bright light. They like to be in the dark. So it's a very simple task, and the animals learn the location of the escape hole. And let me give you another example, if you don't mind. Imagine you are pulling your car into a crowded parking lot. You park, you go into the store. You need to remember where you parked. And I'm sure we all have the experience of not remembering and then going, oh my, where did I park my car, right? So you need to remember where your car is parked. And that requires hippocampus functioning normally. So now imagine you come out of the store and you've forgotten where you parked the car. You have a couple of possible solutions. One is you just sort of randomly walk through the parking lot until you find your car. The other is I'll go up and down every aisle until I find my car. And then when you find it, you go, aha, your hippocampus helps you make a map of where that car is. Now you need to go back in the store because you left your wallet behind and you come out, right? How do you find the car? Your hippocampus has made a spatial map of the location of the car. So it's a very important kind of spatial memory. And it's one of the first kinds of memory, for example, that is seen to be impaired in Alzheimer's disease. So in our rat model, we put them on the platform, we allow them to wander around and find the escape hole, and then we test them repeatedly. If the rat is processing spatial information correctly, it quickly learns to go directly to the escape hole because it has a spatial map of the maze and where the escape hole is. If the rat does not have a good map, he will either wander randomly or might take what we call a serial approach. It would be like going up and down the rows of the parking lot. In other words, they'll go around the edge systematically until they find the hole. Well, what we found was that while the Alexander disease rat could actually learn to find the escape hole, the rat did it in a different way from the normal rats. In other words, it continued to use this random search strategy or a serial search strategy and never developed this hippocampally dependent spatial map of the maze. And so it's apparent that the animals have problems learning something that is hippocampally dependent, and that is this development of a spatial cognitive map. I think that links the areas of brain involved in visual spatial memory, like the novel object recognition, to the spatial deficits that we're seeing, and the interaction of the parietal cortex and the hippocampus.
So while we don't typically subject people to novel object recognition tests or the Barnes maze, you feel that these kinds of tests exploiting normal aspects of rodent behavior reflect processes of learning and memory that are analogous to those taking place in people?
In actual fact, there are Barnes maze, versions of a Barnes maze used in patients with memory problems.
Oh.
They have virtual glasses that allow them to wander either in the same kind of maze with holes that they could go to, or it's a map of a city street, and they have to learn their way through there. And initially, as you would expect, they wander randomly, but eventually they develop a spatial map and they can go directly to a visual world building, okay? So they can develop it. So that's very analogous to the Barnes maze. And then the visual object recognition, people sit in front of a screen and they see objects presented, and then a novel object is presented. If your memory is good, and this is a pretty easy test, but if your memory is good, you'll be able to immediately point to that and say, that's a new object out of all the objects that I've been looking at. So there are analogous kinds of tests that are used in humans and used, as standard part of memory testing. So, I'd like to think that the studies in rodents and the studies in humans are valid ways to study memory.
Let's sum up. Tracy, what do you think the key take-home message from your part of this paper is?
Well, one of the things I've been interested in for a while, and we had a paper come out with Steve Gammy a year or so ago, was just the correlations with diseases like Alzheimer's and Parkinson's really as well, in terms of the profile, and that's in the title, so that's kind of an obvious one. But we're talking about rats that are really young, and even at 8 weeks, that's a young adult, and to see the same profile in these Alexander disease rats at an early age, I think says something, and we'll have to figure out what that is. But I think it ties in nicely too with a paper we've done with Melfini, showing that astrocytes are, at least a subset are, astrocytes are senescent, which is kind of a early aging stage where the astrocytes don't divide or continue to have normal functions, and they're at kind of at a late stage before dying. And so we see astrocytes, senescent astrocytes in our models, but also in the human disease. So how much are those contributing to the overall phenotype or the overall profile of the animal and in the human disease? So I think that was one interesting thing. And then, of course, I think one of the newer items, we saw much of the same types of profiles in our rodent models. This is going back many years now. But the mitochondrial data, I feel, are new, and or at least more striking. The changes are more striking in the rat model. And of course, the rat has a more severe disease presentation compared to our earlier mouse models. So maybe that's an important finding, and I think it would go towards the astrocytes' role in maintaining balance and homeostasis in the brain.
Energy balance.
Yes. One of the questions we've had, is it astrocyte dysfunction in terms of their role in maintaining the brain that's causing Alexander disease, or is it this neuroinflammatory response that's really kind of like a gain of function, and it's this, the cytokines and everything else that are being secreted and the crosstalk with other immune-related cells in the brain that are causing problems? And we still don't have an answer to that, but the inflammatory response comes up first. So that still may be giving us some insight into that question.
And Rob, what's your take-home message?
Well, I actually have three, and one of them is very basic, and that's after all the years that I've been studying animal models of neurological disease, I'm always impressed that simple mutations in single genes can lead to such complex behavioral changes, and in particular can lead to changes in memory and learning. And so something that begins as a mutation in a gene important for astrocytes can lead to a major neurodevelopmental disorder. The other is that the rat model was developed so that we could use it in part to test novel therapeutic strategies. And so now we have additional tools. We can look to see whether or not treatments, whether they're gene-targeted treatments or pharmacological treatments, are also able to have effects on something like memory and learning. And then the last thing is that it seems to me that there have been reports in the literature, mostly case reports, small studies in few individuals, of cognitive problems in Alexander disease and some indication of memory problems, but it hasn't really been studied systematically. And I think that was something that should be done. Because there might be unrecognized or underappreciated cognitive challenges that individuals with Alexander disease may be facing. So those are my three.
All right. Well, thank you again to both of you for coming on today to talk about this paper. That's all for today's episode of Alexander Disease Research Update. Thanks for listening, and thank you to Rob and Tracy for joining me today. Our theme song was written by Charlie Allenson, and support for these podcasts has come from the Barrett Riddle family. I'm Albie Messing. See you next time.